Dear student, myself Jiten Verma, Assistant Professor, Government College, Kota. Today I will teach you root apical meristem. ये टॉपिक बेसिकली एमएससी सेमेस्टर थर्ड के लिए है और उसी डिटेल के हिसाब से तैयार करा गया। हालांकि सेम चीज़ यूजी क्लासेस में भी अच्छी यूजफुल रहेंगी। रूट एपिकल मेरिस्टेम। रूट एपिकल मेरिस्टेम जो है आर द सब एपिकल रीजन ऑफ एपिकल पोर्शन ऑफ द रूट के सब एपिकल पोर्शन होता है जहाँ पर कि मेरिस्टेमेटिक सेल्स होती हैं। ये विभिन्न प्रकार के इंटरनल टिश्यूज़ रूट्स के बनाते हैं, जिसे हम डायग्राम में देख सकते हैं कि इस तरह से इसका रूट एपिकल मेरिस्टेम का क्षेत्र होता है। ये हमेशा सब टर्मिनल रीजन है क्योंकि ये कवर्ड होता है रूट केप के द्वारा। ये वाला पोर्शन रूट केप है। इसमें किसी प्रकार के नाइदर एनी लेटरल अपेंडेजेज और ब्रांच और देयर ग्रोथ जो इट इज स्मॉलर देन द रूट शूट अपेक्स से छोटा होता है। द सेल्स ऑफ द रूट एपिकल मेरिस्टेम कंसिस्टेंटली डिवाइड एंड देयर एक्टिविटी इनेबल्स द रूट टू ग्रो इन पॉजिटिवली। जमीन की ओर ये ग्रो करता है और फोटोट्रॉपिक के नेगेटिव डायरेक्शन में है, लाइट के अपोज में ही ग्रो कर रहा है। इस तरह से इसका क्षेत्र ये दिखाई दे रहा है हमें। देखिए ये रूट कैप दिखाई दे रहा है, यहाँ पर क्विसेंट सेंटर है। इस जगह मैक्सिमम रेट ऑफ सेल डिवीजन होगी। ये सेल डिवीजन से यहाँ नहीं होता है आगे। ये इलोंगेशन जोन है। पहले मेरिस्टेमेटिक जोन रहेगा, जैसा कि आपने पहले माइटोटिक डिवीजन की स्टडी में भी सेल मतलब रूट्स काटी होंगी, ओनियन रूट से करा होगा। जब भी हम देखते हैं कि ये वाला क्षेत्र जो है ये मेरिस्टेमेटिक है, ये इलोंगेशन जोन रहता है और इसके बाद का क्षेत्र ये है जो मैचुरेशन जोन कहलाता है। इस तरह से इसको हम टीएस में भी देख सकते हैं रूट्स को, कि इस तरह से ये रूट का टीएस स्ट्रक्चर दिखाई देगा, क्रॉस सेक्शन जिसको हमको। और ये एलएस जो अभी हमने बताया ये है, जैसे रूट केप हो गया, जोन ऑफ सेल डिवीजन, जोन ऑफ इलोंगेशन, जोन ऑफ मैचुरेशन। और इस तरह से यहाँ से एंडोडर्मिस निकल रही है, रूट हेयर्स है, यहाँ रूट हेयर निकल रहे हैं। ये एपिडर्मल फर्स्ट लेयर है, देन ग्राउंड टिश्यू, वास्कुलर टिश्यू, ये ग्राउंड मेरिस्टेम, प्रोकैम्बियम, प्रोटोटर्म, ये क्विसेंट सेंटर है जैसा अभी हमने अगले डायग्राम बताया। देन इस पोर्शन को ये एपिकल मेरिस्टेम ये कर रहे हैं। ऑर्गेनाइजेशन ऑफ रूट अपेक्स। एपिकल सेल्स ऑफ द प्राइमरी रूट्स आर मेरिस्टेमेटिक इन नेचर एंड दे अटेन द कैपेबिलिटी टू डिवाइड एज सून एज द एम्ब्रियोनिक रेडिकल फॉर्म्स. The cells of the root apical meristem have the bigger and prominent nucleus and have dense cytoplasm. Either lack vacuoles or vacuoles are very small. Cytoplasm are very dense. These cells are neither either ellipsoidal or polygonal in shape. Their cell wall is the thin and uniform. They divide to form the cells of the mature root system. Root cap cells are fully matured cells which develop from the dermatocalyptozone. Tissue system comprised of the epidermis, cortex, vascular, etc. Type of root apex. Root apex has been classified into the 5 types on the basis of the method of origin of the root cape and interrelations among histozoan and primary tissue. These diagrams show the various 5 types. Type of the first is the A type, and second found in the gymnosperm, C found in the dicotyledon, and D type found in the monocotyledon. A. First type. In this type of the root apex, the single solitary cell is found in the terminal position. Single. This terminal cell produces root cape and other tissues of the roots. Second type of the root apex. Single layer of meristem cell found in the epidermis, cortex, vascular tissue, and root capes. These develop on this type of the root apex. Such type of the root apex organization found in the ranunculation. The second type. This one. The second type found in the. And third type is the most of the gymnosperm. 2 groups of the initial cells are present in the root apex. Inner portion produce the plerome, while the outer portion produce peribleum and root apex. This is the C type found in the dicotyledon, and B in the gymnosperm. Fourth type, this type found in the root apex organization found in the rest of the angiosperm. In such type of the root apex, meristematic cells organized into 3 layers. The outermost layer gives rise to dermatozoan and root cape. Middle layer give rise to peribleum and inner region give rise to the plerome. This one is the diagram. This is the found in the monocotyledons. Root apex region and their function. Meristem of the root apex found in the vascular cryptogams, pteridophytes. Scope recognize this type is the type A. This type F. Theories related to the root apex organization. Multiple theories has been proposed to understand the structure and organization of root apex. Basically, three theories are recognized to explain this type of the organization in the root. First is the histoapical cell theory, and second is the histozoan theory, and third one is the Corpus Cape theory. First, apical cell theory. This theory was proposed by the Hofmeister in the 1957, and later this was elaborated by the Nageli. According to this theory, there is a tetrahedral cells in the root apex. The tetrahedral cells in the root apex which divide into three planes, producing different tissues of root. Division in the basal plane of tetrahedral cell give rise to the root cape, and LS. This theory acceptable for the some of the pteridophytes like the Polypodiaceae, Ophioglossiaceae, Equisetaceae and Azulaceae because of the differentiation and formation of the various tissue and related regions in this plant groups. According to Gunning, Ital and Hardman, the apical cells of the root apex in Azolla divides for about 55 times to initiate the root growth. However, this theory failed to explain the structure and organization of root apex in the case of the spermatophytes like gymnosperm and angiosperm, because they are a group of meristematic initial cells in the gymnosperm and angiosperm. The case, the apical portion of the root apex which actually divide and differentiate to form the different tissue of the root. Another theory given by the Henstein 1970s. This is called the histozoan theory. This diagram shows the LS of the meristem. This is the quiescent center, and this is the ground meristem, procampus. This showing by the Henstein. He proposed this theory after studying the shoot and embryo of the meristem, that the meristematic cells of root apex are made up of the three layers. Exactly similar to the root apex epidermis. He identified presence of the three regions in the shoot and root also. This he found three regions. Then LS of root apex depicted three regions are dermatozone, peribleum and the pleron. Pleron is the central meristematic part of the apical meristem of the root. This form of the stereo tissue some parts of the ground tissue like pericycle, pith rays and medullary rays and pith. These three layers were collectively known as the histozoan. Another important theory is the Corpus Cape theory. This diagram shows the Corpus Cape theory's explanation. This theory was proposed by the Scoop 1917. According to this theory, the cells of the root apices are divided into the two elements. The first division is of transfer type, resulting in the two cells, out of which one divides anticlinal called as the T division. In some of the portion of the root apex, especially the middle portion, T is seen upright, while the rest of the region inverted T is seen. This type inverted T is seen. When the T is upright, then it is directional toward the apical portion, but when the T is inverted, this direction opposite to the apical portion. Scoop named upright as the corpus and the body, while inverted T are called the capi. This theory is equivalent to the tunica carpus of the shoot. These diagrams show the this type is the inverted T and this one is the T. Unlike the shoot apical meristem, the root apical meristem which flank the quiescent center produce cells in the two dimension at the periphery and together produce most of the cells in an adult root. He recognized these cells as constituent of the promeristem. As it terminus, the root meristem is covered by the root cape which protects and guides its growth trajectory. Cells are continuously shed off and outer surface of the root cape. Root apical meristem and tissue pattern are established at the very embryo stage, in that case of the primary root. The cells of the quiescent center has lesser DNA, RNA and protein content. These cells have lesser number of the endoplasmic reticulum and mitochondria. Nucleus and nucleolus are smaller in size. The quiescent cells are characterized by their low mitotic activity as they maintained at the G1 S checkpoint in the cell cycle. The cells of the quiescent center remain inactive till the peripheral cells are active stage of the division. The cells of quiescent actively divide when they are exposed to the damaging dose of the X-ray, while other meristematic cells do not show such response. Histones theory and Corper-Kappa theory successfully explain the organization of the root apicase, except the presence of an independent calyptrosome and 4-cell layered root in the monopod. The activity of the meristematic tissues enables root to grow and the zone of elongation and root head zones grow. Root cap protect the root from the damage proposed by the positive geotropic. The cells of the root cap are impermeable to water, while the meristematic cells of the root apicase have the capability to absorb water along with their capacity to divide. The cells of the zone of elongation do not efficiently absorb water, while actively absorb the loss of mineral elements. The cells of the root head zones are most permeable to water. The cells of the zone of maturation are lesser permeable due to the deposition of lignin and sebaceous. Thank you very much.